Okay. Can you hear me?
Yes.
Yeah. Good morning. This is a session of Microbiology and Immunology 1. I'll chair this session. My name is Shin Nakamura from Nova Southeastern University in Florida. Another chair is Dr. Snack Sarkar from Ashland. Dr. Sarkar, could you please introduce yourself?
Are you ready for your first
Yeah.
presenter?
Okay.
Yep.
First presenter is Dr. Miki Aramiro from Indiana University School of Dentistry. Can you share the screen?
Yes, can you see that all right?
Yes.
Okay, perfect.
Okay, go ahead.
All right, thank you. Yes, my name is Miki Jaramillo, and I am a third-year dental student at the Indiana University School of Dentistry. I will be presenting on soluble ACE2 expression in symptomatic COVID-19 patients. My work was conducted with Dr. Srinivasan and Dr. Thivalakakis. Angiotensin-converting enzyme, or ACE2, is the most significant receptor for SARS-CoV-2. As an enzyme of the renin-angiotensin system, it is a modulator of systemic and local inflammation. It is expressed on epithelial cells throughout the body, including in the oral mucosa and salivary glands, suggesting a connection to the common oral symptoms associated with COVID-19. To cause infection, SARS-CoV-2 binds via the S1 subunit of its spike protein to ACE2 on the host cells. In the figure, ACE2 is indicated in green and the incoming viral particles are orange. The spike protein is then cleaved between the S1 and S2 subunits by the transmembrane serine protease 2, allowing the remaining viral particle to fuse with the host cell and begin replication. The metalloprotease ADAM17 cleaves the extracellular domain of ACE2, resulting in the release of its soluble form. TLR4 on oral epithelial cells may facilitate the interaction between ACE2 and SARS-CoV-2, which would induce inflammation and activate macrophages, leading to the common oral symptoms of mucosal erosion and ulcer formation seen in COVID-19. The soluble ACE2 form can act as a decoy receptor by saturating the binding sites on SARS-CoV-2, thus modulating disease expression. Because of this, soluble ACE2 has been suggested as a possible treatment regimen for SARS-CoV-2. Our project sought to assess the sensitivity and specificity of saliva as a collection method for soluble ACE2. Additionally, we sought to evaluate the difference in expression of soluble ACE2 of symptomatic SARS-CoV-2 infected patients compared to healthy individuals. Saliva samples were collected from 30 symptomatic COVID-19 patients that were admitted to the hospital and diagnosed using a reverse transcriptase PCR assay from nasopharyngeal swab samples. These were compared to the control group, which consisted of archived saliva samples from healthy individuals of comparable ages. The saliva samples were analyzed using a PCR assay and a binding assay. The PCR assay amplifies the RD, RP, and N genes of viral particles. This is used to calculate the cycle threshold, which is the number of PCR cycles necessary to produce a fluorescence above the background. A standard curve is applied to translate cycle threshold into viral load. The binding assay measures the binding activity of soluble ACE2 in saliva to a plate pre-coated with the SARS-CoV-2 spike protein. Anti-ACE2 antibody was added and its absorbance measured. This was used to calculate the binding activity according to the equation below, where OD is optical density and the slope was calculated from a linear regression. In this table is a breakdown of the patient cohort by age, sex, symptoms, and comorbidity. Most patients in the cohort were over 50 years old and female. The most common presenting symptoms included shortness of breath and coughing. Most of the sample also had at least one pre-existing comorbidity. As shown in the blue graph, of the 30 hospitalized COVID-19 patients, 5 samples were discarded due to insufficient volume. 7 of the saliva samples tested negative for the SARS-CoV-2 in the Abbott PCR assay that we performed. 2 of the saliva samples were inconclusive, and the remaining 16 were positive. As shown in the green bar graph, the average cycle threshold ranged between 13.6 to 30.47 for these positive samples. Based on the CT values, we determined the viral load, as shown in the orange bar graph. This ranged between 1.6 to 109 viral copies per milliliter for samples 2 through 12, 14, and 15. There was a moderate correlation between the viral load and the mean CT value with an R squared of 0.66. The CT value for sample number 2 was beyond the limit of detection. However, the viral load for the sample was positive, indicating that the PCR assay for the sample was a false negative. The salivary levels of soluble ACE2 were determined by the binding assay. In panel A, we see the soluble ACE2 binding levels averaged for the COVID-19 group and the control group. Saliva of symptomatic COVID-19 patients exhibited significantly lower soluble ACE2 as compared to the saliva of healthy control individuals at a p-value of less than 0.05. In panel B, we see the individual soluble ACE2 binding activity levels plotted versus their respective CT values, demonstrating an inverse relationship with a moderate correlation at a Spearman's rho value of negative 0.48. There were some discrepancies between the nasopharyngeal swab tests that the patients received upon admission to the hospital versus the PCR assay run on the collected saliva. This could be attributed to any of the following. The time of sample collection with respect to the patient's admission. Typically, saliva was collected 24 to 48 hours after the nasopharyngeal swab, independent of the patient's course of infection or the initiation of treatment therapies, and the difference in the sensitivity between the nasopharyngeal swabs and the saliva. The study was also limited by the small sample cohort and by the presence of comorbidities. Our study did show that COVID-19 positive saliva samples exhibited significantly lower soluble ACE2 as compared with the saliva samples from healthy individuals. This may be attributed to the potential protective role that soluble ACE2 serves against SARS-CoV-2 by functioning as a decoy receptor. However, the amount of soluble ACE2 in saliva correlated weakly with the SARS-CoV-2 viral load. Currently, we are working to assess inflammatory markers such as toll-like receptors to evaluate a possible correlation with soluble ACE2 levels in patients with an active COVID-19 infection. Our preliminary data shows that TLR4 levels are significantly higher in COVID-19 patients as compared to our healthy control group. This may be attributed to TLR4's potential role as a receptor for SARS-CoV-2 viral particles. In conclusion, our data suggests that reduced soluble ACE2 in saliva could represent a biomarker for severity of COVID-19. Thus, soluble ACE2 may be used as a prognostic indicator in coronavirus infections. As part of our future direction, we will be evaluating soluble ACE2 levels in a larger population and including asymptomatic COVID-19 patients to validate. Additionally, we are working to assess pattern recognition receptors to evaluate a possible correlation with soluble ACE2 levels in patients with an active COVID-19 infection. And I'd like to thank Dr. Srinivasan and Dr. Bruce Needy at Indiana University. Thank you.
Thank you. Thank you for your great presentation. Do you have any questions?
I have a question. Can I ask one now?
Yeah. Please.
Did you look at, I know you isolated RNA and did RT-PCR. Did you do RT-PCR for ACE2 as well?
We just focused on soluble ACE2 in the saliva, but we did run our reverse transcriptase real-time PCR assay on that saliva for soluble ACE2.
For the ACE2 gene, for the ACE2 expression.
Yeah, for the soluble form, yes, for ACE2. Mm-hmm.
Okay. Was the graph you were showing, was that protein or RNA? That's what I'm trying to understand.
This one?
Yeah, that's binding activity. Okay, because you didn't show any RNA. So because the reason I ask is that we published about a year ago the same thing, but we did only RT-PCR, which shows that ACE2 goes down in COVID patients in the mouth.
Mm-hmm.
So
very interesting. I'll have to look for that article.
I can send you an email. I'll send
yeah, that'd be great.
Sure. Okay, but you say you also did qPCR for ACE2.
Yes, yeah, we did do qPCR.
And do you have a similar result?
Yes, we did have lower soluble ACE2 in saliva. Yes.
ACE2. Okay, good. Good to know that our results aren't wrong.
Thank you. Time is coming. Let's move on to the next speakers.
Thank you.
Thank you again. The next speaker is Dr. Nicole June from the University of Detroit Mercy School of Dentistry. Could you share the screen? Okay, go ahead.
Hi, good morning. My name is Nicole Jun and I'm from Detroit Mercy. I'm a DS3 student and I will be presenting today on the pre-colonization of oral probiotics to prevent strep mutans biofilm formation. So dental caries is the most prevalent chronic disease worldwide. It's widely accepted that the bacterium Streptococcus mutans is the main contributor to tooth decay due to its ability to produce high levels of lactic acid in the presence of carbohydrates, thus leading to enamel demineralization and caries formation. Fortunately, dental caries is largely preventable. New discoveries in modern medicine have established the use of probiotics, which are live microorganisms to confer various health benefits, traditionally associated with gut health, but more recently, studies have proposed the use of probiotics to prevent strep mutans mediated caries formation. Numerous oral probiotic products are on the market, such as probiotic toothpaste and tablets, and these advertise decreased tooth decay. The aim of this study was to investigate the potential benefits of probiotics in the oral cavity, and with the help of another student, my PI and laboratory technicians, we were able to conduct a set of experiments to determine the various effects of oral probiotics on strep mutans biofilm formation and viability. We investigated several commercially available products. Periobiotic was the first product and it contained Bacillus coagulans. Biogaia contained Lactobacillus reuteri, and Vigis 101 is a strain of Lactobacillus paracasei. The final product in our study was Probiora, and Probiora is a mixture of 3 Streptococcus species, Streptococcus oralis, Streptococcus rattus, and Streptococcus uberis. All of these probiotics have been previously studied shown to produce compounds that may challenge the growth of strep mutans. To study the impact of these products on strep mutans biofilms, we first grew the probiotics and strep mutans alone to determine their independent biofilm growth patterns. Then, in additional wells, we grew the probiotics and strep mutans together, and we predicted that this would have a marginal inhibitory effect on strep mutans. Finally, we grew the probiotics for 3 hours to condition the medium prior to adding strep mutans. We predicted that these wells would have the most dramatic impact on strep mutans biofilm formation. And to analyze the growth of the biofilms, we used several different assays. The first was a crystal violet stain. In this slide, you can see a graph comparing the first 3 products, the Periobiotic, Biogaia, and Vigis. By setting the biofilm formation of strep mutans alone to 100%, we established a baseline comparison to measure the biofilm density, and this was done with a crystal violet stain. It was used to determine the biofilm density, and on the y-axis, we have the percent of biofilm growth relative to strep mutans. On the x-axis, the probiotics are listed. Independently, we can see that the probiotics were unable to form dense biofilms. This would be the Periobiotic and Biogaia. And in the wells that were plated simultaneously with strep mutans, Lactobacillus paracasei showed a statistically significant reduction in biofilm production when compared to the baseline growth of strep mutans. However, contrary to our prediction, the Biogaia showed a considerable increase in the biofilm production. In the final section of the graph, we have the probiotics that were grown for 3 hours prior to the addition of strep mutans. In this experiment, when given the ability to pre-colonize the well, both the periobiotic Bacillus coagulans and Lactobacillus paracasei showed a statistically significant reduction in the biofilm density. As our goal for the project would be to inhibit enamel demineralization and caries formation, we also measured the biofilm pH. Enamel demineralizes at a critical point of below pH 5.5. However, none of the probiotics were able to reduce the acidity of the biofilms relative to strep mutans alone. In this graph, we assessed the effect of the Probiora, which was the mix of the 3 Streptococcus species. We isolated the 3 species and then tested them all together. Each probiotic component independently was shown here, and you can see that the Strep oralis and the Strep radus were, Strep ubris were unable to form dense biofilms. In here, the simultaneous section of the graph, we can see that the combination of the three species was able to inhibit biofilm production. Additionally, when plated prior to the inoculation of Strep mutans, it was able to inhibit it as well. Notably, though, when Strep ubris and Strep oralis were plated prior to the inoculation of Strep mutans, they were able to reduce the biofilm density. And with previous research, it was shown that both of these species produce hydrogen peroxide, which may contribute to its ability to inhibit the activity of Strep mutans. And despite reducing the thickness of the biofilm formation in many of the cases, in no case was the probiotic able to reduce the acidity of the biofilm. To assess the ability of the probiotics to secrete antimicrobial compounds, we had a plate competition assay. So we would spot Strep mutans and let it grow alone and see how it would be able to form a robust colony. But when plated nearby a probiotic, the growth of Strep mutans was inhibited. Here we can see the growth of Strep mutans alone, and it was able to form a robust colony. Here, side by side, these were the first three products that I had discussed with the Bacillus coagulans, Lactobacillus reuteri, and the Lactobacillus paracasei. We let the probiotics grow for 48 hours to monitor the production of the diffusible antimicrobial compounds. And here we can see that the growth of Strep mutans was inhibited. And the four images below, these were the products from the Probiora, which were the 3 Streptococcus species, and all of the probiotics had an ability to inhibit Strep mutans colony formations by varying degrees. So in conclusion, we determined that probiotics, Bacillus coagulans, Lactobacillus paracasei, Streptococcus oralis, and Strep ubris were able to inhibit Strep mutans biofilm formation. Additionally, the plate competition assay showed that all probiotics tested had the ability to hinder Strep mutans growth through the secretion of diffusible antimicrobial compounds. Our findings led to the idea that the probiotics tested had the ability to reduce Strep mutans biofilm formation. However, of particular interest is the fact that even when Strep mutans biofilms were reduced, the pH was not less acidic, and this is due to the ability of several probiotics to make lactic acid on their own. So it is a question to see how substituting one acid producing bacteria with another would be beneficial for caries reduction. Although the initial results of our study had shown promise to conferring oral health benefits, additional studies were necessary to determine the composition of the biofilms and the mechanism of action by which the probiotics were inhibiting Strep mutans biofilm formation. Our future experiments will include determining the species composition within the mixed biofilms via quantitative PCR and using mass spectrometry to determine the compounds that each probiotic was secreting to inhibit Strep mutans growth. And I would like to say thank you to my research professor, Dr. Krikonis, and those who helped me with our project, Ms. Olivia Erminger, Carmen Rucker, Laura Young, Sarah Fletcher, and Dr. Thompson. Thank you.
Thank you for your interesting presentation. Do you have any questions?
Hi, can I just ask a question? Thanks for that talk. Can you just tell us what medium you use to grow the biofilms, please? Do you have sucrose in there?
Yes, we grew the biofilms with artificial saliva and sucrose.
Okay, be interesting to see what happens with the competition if you don't have sucrose, or at least not present constantly, because it wouldn't be always in the mouth.
Mm-hmm.
Thanks.
No problem.
Thank you. Okay, time is coming. I will move on to the next speaker. Thank you again. Third speaker is Dr. Deva Seferovic from College of Dental Medicine, Midwestern University. Could you share the screen?
Yes.
Thank you. Go ahead, please.
Perfect. Hi, my name is Dwar Seferovic. I'm from Midwestern University. Thank you for everyone for being here. Before I get started, I want to thank the research team at Midwestern University. I wouldn't be able to do this research without them. But I'm here today to talk to you about the antimicrobial capacities of a silver nanoparticle containing tooth gel. So some background. When we hear about silver in medicine, a lot of the times we head straight to snake oil salesmen. But there's a large body of research showing that silver nanoparticles actually are extremely antibacterial. And specifically in the dental world, there is research of their use in prosthetics such as RPDs and dentures. We're starting to see a lot of research of their use in implants as well. What we do know about silver nanoparticles is the smaller the particle size, the more of an antibacterial property it does exhibit. And so when we're looking at particle size, we're looking for anything that's smaller than 7 nanometers to be an effective antimicrobial agent. And the gel that we did study, the Silver Biotics Tooth Gel, contains silver sol, which is a nanoparticle that is 3 to 5 nanometers large. So what was our objective of the study? Our objective of the study was to measure the bacterial reduction of a commercially available silver nanoparticle gel and the reduction of the other active ingredients in the gel. A lot of these products that are marketed on online retailers or other sites are marketed as silver nanoparticle gel antibacterial, but they don't state on the front at least that there's other active ingredients such as peppermint oil and xylitol, which there is, again, a large body of research showing that they're antibacterial or bacterial static and bactericidal. Specifically, we were looking at strep mutans when we're doing this research. So what was our method? We grew strep mutans to an optimal density of 0.6. Then we took 1 millimeter of that and we put in 9 millimeters of broth. We took about 0.6 mLs of the gel, which we deemed as the recommended dose of the gel. Took the MSDS form, we were able to extrapolate how much peppermint oil was in there and how much xylitol was in there. So it was 13.6 microliters of peppermint oil and 1.375 grams of xylitol, which was in the crystal form. We then vortex that, and then we picked time intervals of 2, 4, and 6 hours, and that's why our samples were incubated for that long. So for our first time interval, we took 1 millimeter of our treatment groups. We put them in 9 millimeters of broth to do our dilutions. We did a factor of 1 and a factor of 3 for the first time interval, used a spiral plater to plate that. Then we let them incubate for 48 hours, and then we use a colony counter to get our CFU units. At the 4 and 6 hour intervals, we took 100 microliters and put in 10 millimeters of broth and did a dilution of a factor of 2, 4, and 6. Then again, we used the spiral plater, incubate 48 hours, and then we used our colony counter to get our CFU units. So this is an example of peppermint oil at 6 hours at the dilution of a factor of 2. So when we did get our data and we were able to calculate our CFU units, we took the first and last time interval, ran an ANOVA test to see if there's interaction between time and the treatment groups. And then we also, as you can see, we use just plain blood agar plates. So what were our results? Our results show that there was a significant, statistically significant effect for the treatment. There's really no interaction between time and the treatment. The peppermint oil did show that it significantly lower CFU units, and you'll see on the next graph how much lower. And then even though there was a reduction with the gel and the xylitol, over time, it was not statistically significant. So if we look at our graph here, the control is in blue. We can see that the gel and the xylitol do have some reduction, but again, they were not statistically significant. And the peppermint oil had a significantly lower colony factor unit than any of the other treatments. So why do we think this is happening? we honestly think in the gel, the peppermint oil is playing the largest role. And the other active ingredients are having some effect, but we think mainly the issue with the gel is that the constituents of the gel, that the fact that it is a gel is actually inhibiting the silver of exhibiting its antibacterial properties. So if we would redo this experiment or for further research, we'd probably use a mouthwash to try to take that element out. Some other limitations were the time to complete the intervals. Honestly, trying to use as many plates as we did and trying to get them done and not inhibit the growth curve of the bacteria was extremely difficult. It took us a few runs to get efficient at, and me and the research team were able to actually figure that out, and it worked well. We also don't know the effect of the treatment is having on the culture. Is simply the peppermint oil having such a massive die-off and such an massive antimicrobial property because it's maybe changed the pH of the broth, or other factors like that, which we can't really test for, which we really didn't test for, and it would be difficult to test for. Also, all the research we're doing is in vitro. It was done on the benchtop, and it does not simulate a lot of the oral conditions that might actually inhibit peppermint oil or might improve the performance of the gel. So our results showed that the Silver Biotics gel has is antimicrobial, but we do think the peppermint oil is playing this most significant roles, and that this in vitro study does justify the use of it, of the gel in a clinical study. And I want to acknowledge the Dean's Fellowship at Midwestern for allowing me to do this research, and SCADA for helping me present here. Also, I want to acknowledge again the research team. They were unbelievably wonderful in all aspects, and I want to thank all of them. And questions.
Thank you for your great presentation. Do you have any questions? Sorry. Sorry, the connection is not good.
Yeah. I had a question about the silver, the copper, and the nanoparticles.
Yeah.
With the nanoparticles, what concentration of silver was used in the gel for the silver nanoparticles? Do you know?
So that was actually difficult to find. We weren't able to actually extrapolate for that from the MSDS form, and that was the issue we're having with the gel. The MSDS form did not specifically state the concentration. So what we were looking at was our main focus on this study was these products are commercially available, mm-hmm. and the silver particles aren't well studied in uses of like prophylactics or prevention. So we just took whatever the concentration was. We weren't really able to extrapolate that out of the gel, and it's something that we would do in the further try to figure out the concentration of that.
Okay, my lab is working with silver nanoparticles right now, and we're trying to find the correct concentration for antimicrobial effects. So I was just curious. Treatment, or if there wasn't a concentration that has been necessarily set for the silver.
You keep breaking in and out.
I'm so sorry. The Wi-Fi.
You want to send me a chat maybe, and I can.
But thank you. That would be perfect. Awesome. Thank you. Sorry.
Thank you. Let's move on to the next topic. Thank you, Petrovich.
Thank you.
Next speaker is me. Let me share the screen. Can you see? Okay. I'll begin my presentation. Porphyromonas gingivalis, PG, is implicated in the systemic disease associated with periodontal disease. PG DNA and gingipain were detected in human brain. However, it remains elusive how PG can be transported to such a remote tissue from periodontal region. Therefore, we focused on efferocytosis process. Efferocytosis plays an essential role in the removal of apoptotic cells, avoiding inflammation. Dying apoptotic cells express phosphatidylserine, PS, on their cell surface as a litmus signal. Macrophages recognize the PS via the bridge proteins. Among bridge proteins, we focused on JAS6. JAS6 is expressed in epithelial tissue under normal condition. Also, JAS6 and LY6 positive cells, you mean macrophages, is upregulated by PG infection. And interestingly, PG produces unique sphingolipid PGDHC. This function is still unclear, but the structure of PGDHC is similar to PS. So we assume that macrophages recognize PGDHC via JAS6 as well as PS, and non-inflammatory phagocytosis is induced, and macrophage migration might be promoted to revert PG. The aim of this study is to investigate the effect of JAS6 mediated recognition of PGDHC on macrophages, phagocytosis, and migration. We conducted a series of experiments with this strategy. We employed PGW83, a wild type, and SPT knockout strain. SPT is an essential enzyme to produce PGDHC. I mean, SPT knockout strains doesn't have PGDHC. We investigated, firstly, we investigated the binding of PGDHC to annexin 5. Annexin 5 can bind to PS. As a result of fluorescent histochemistry, PGDHC appeared to bind annexin 5. In short, PGDHC has functional similarity to host PS. Next, we examined binding activity of PGDHC to JAS6 with BLI analysis. In BLI analysis, we can evaluate binding affinity using biosensor. Consequently, JAS6 bound to PG wild type, but not to PGSPT. JAS6 can bind both PGDHC and host PS. Also, we measure the production of inflammatory cytokine. After PG stimulation, PGSPT promoted TNF-alpha production. And addition of JAS6 diminished TNF-alpha production in 2 hours. In short, JAS6 decreased the production of cytokine at the early stage of inflammatory reaction. Furthermore, we evaluated bacterial phagocytosis in 2 hours with flow cytometry using fluorescent labeled PG. The results showed JAS6 didn't affect the phagocytosis of macrophage. On the other hand, we evaluated the viability of PG in 2 and 24 hours using agar plates. The results showed survival of PGSPT decreased compared with PG wild type, and the survival of JAS6 treated PG increased compared to other groups. In short, PGDHC might be engaged in survival of PG, and JAS6 might promote the PG survival through possible interaction with PGDHC. Finally, we conducted a migration assay. We evaluated the migration of macrophage containing PG. As this result, the exposure of PG increased in macrophage migration, and Gas6 treated PG wild type increased macrophage migration compared with the other groups. The phagocytosis of Gas6 treated PG may promote the migratory capacity of macrophages. As mentioned earlier, we've shown these results. To sum up, the survival of engulfed Gas6 treated PG might increase by interaction with PGDHC. And PGDHC may play a role in Gas6 mediated non-inflammatory phagocytosis of PG by macrophages and promoting their migratory function. In conclusion, we suggested that Gas6 mediated recognition of PG via PGDHC induces non-inflammatory phagocytosis, which in turn promotes the migratory function of macrophage. In future, we need further investigation about the molecular mechanism of phagocytosis and migration via PG and Gas6. That's all. Thank you for your attention. Do you have any questions?
I was just curious. I'm trying to look back over the slides. Could you back up the slides to show me about how the how you showed that the PG was not killed in the macrophages that were that had the that were taken up by through Gas6? Because that's the curious part to me, is that when they're taken up, that the all right, it's okay. That's so they survive within the raw cells. Okay, that's really interesting.
Thank you.
I'm trying to think of how to stimulate the macrophages to make them kill them.
Yeah, well, we need to investigate about the molecular mechanism.
Very interesting. Thanks.
Thanks. Okay, thank you. Let's move on to the next presentation. Next speaker is Dr. Shanak Sarkar. Dr. Sarkar, can you share the screen?
Yep, I'm sharing my screen. Can everyone see it and can everyone hear me?
Okay, go ahead, please.
Perfect. So first, I'd like to start with an apology. When Shin was introducing me, I was speaking into the mic, which was on mute. So my name is Shaunak Sarkar. I am a senior staff scientist at Ashland Incorporation, which is a specialty chemical company. We are located in Bridgewater, New Jersey, and our job is, if something is working, we provide specialty chemicals to make it work better. And that brings me to the topic of my presentation today, using polyvinyl methyl malic acid polymers to enhance the deposition and retention of cetylpyridinium chloride. So as a background, as you might be familiar, cetylpyridinium chloride, or CPC, is a cationic quaternary compound, which is pretty well known as a broad spectrum antimicrobial agent and is prevalently used in mouth rinse, sprays, toothpastes. And at a higher concentration, 0.045 to about 0.1%, CPC has been claimed to effectively stop or work against gingivitis or plaque. And at lower concentrations, it's known, or it has been claimed, to control halitosis in mouth rinses. One issue with CPC is, albeit being a very potent active, antimicrobial active, it's also very water soluble. And that might affect the deposition and retention, and in turn, the long-term antimicrobial activity of CPC in the mouth rinse formulations or other oral formulations. So our objective was to investigate, can we elongate that time of retention and activity of CPC in the oral cavity using some special type of polymers that we have. So the theory is in a mouth rinse, CPC is used at 0.02 to 0.07%, which turns to 200 to 700 ppm. But since CPC is a very potent active, the effective level that might be required is probably much lower. And one of the reasons why high level of CPC is used in the mouth rinses is because it washes off very easily. So our idea was to use something like a molecular sponge that will hold on to the beneficial CPC and help it deposit in the oral cavity, and then over time, keep on releasing the effective level of CPC, thereby allowing the antimicrobial activity to be persistent over an extended period of time. So at Ashland, we have something called Gantrose chemistry, which is a polyvinyl methyl malic acid copolymer. By itself, these are known to be extremely substantive to biological surfaces, especially to enamel, gum, and mucosa. And a specific reason for that is the alternating AB structure of the carboxylic and the methoxyfunctional groups. These are generally like high molecular weight polymers with a flexible polymer chain, which further facilitates the interaction, spreading, and entanglement on the enamel and other mucosal tissues. Besides that, the Gantrose S97, which is the highest molecular weight variant of this polymer, in a neutral solution is inherently anionic. So it can form a reversible ionic complex with an active like CPC, which is inherently cationic, providing benefits that, since it itself is pretty substantive to the oral tissues, it might deposit itself and bring in the CPC with it, thereby providing good retention and deposition. And since the ionic complex is reversible, it can keep on releasing CPC for the antimicrobial benefits. So the first thing we needed to prove is that Gantrose is not hampering the antimicrobial potency of CPC. To do that, we resorted to MIC, or minimum inhibitory concentration tests. Solutions with and without Gantrose and CPC were serially diluted and inoculated with strep mutant cells. And MIC was determined as the minimum concentration of CPC that inhibited the growth of the streptococcus cells. These are all in vitro studies that I'm going to say, and the results showed that, as I said, CPC is pretty effective. The MIC was found to be as low as 0.6 ppm, and adding the polymer, which is supposed to form the ionic complex, did not alter the minimum inhibitory concentration. So now that we know that adding the polymer did not affect the potency of CPC, we needed to prove its deposition and retention benefits. So to simulate a soft tissue, we chose bovine collagen, which was again treated with CPC solutions with and without Gantrose. Then they were dried, air dried on the filter paper, and we did like ATR-IR spectroscopy of the bovine collagen, compared that with the untreated bovine collagen, and to understand the differences between these samples and the contribution coming from CPC to these samples, we did a principal component analysis. The graph on the right-hand side shows the results. The x-axis arises from the contribution of the bovine collagen background material. Y-axis arises from the contribution to the spectra coming from CPC. And a quick, easy way to read this plot is data point that is furthest away from the x-axis contains higher concentration of CPC. It does not matter whether it's in the positive side or on the negative side, it's the absolute distance that matters. And in that respect, we see that the bovine collagen that was treated with CPC plus Gantrose solution had more contribution of CPC than the bovine collagen that was treated with just a CPC solution. So that kind of like tells us that, at least in the in vitro study, the Gantrose S97 polymer is helping in deposition of CPC at a higher level on the soft tissue analog. For a hard tissue analog, we used another technique called quartz crystal microbalance with dissipation, where the deposition and retention of something is measured as a change of frequency of an oscillating quartz crystal. If something deposits, frequency goes down. If the material that is depositing is globular, then the technique also measures dissipation. So what we did is like we took a hydroxyapatite coated quartz sensor, and then we treated them with CPC solution and CPC plus Gantrous solution. So let me explain what the graph says. There are three zones in this graph. First is the water baseline, and then the treatment, which is either the CPC solution or the CPC plus Gantrous solution flown over the sensor. And then there is a water wash part where we flowed water at double the flow rate than the previous two sections to simulate a vigorous washing. So the top graph is what happens when just CPC is flowed over the solution. We do not see a significant change in the frequency line, which are the set of blue lines, which means only a very small amount of CPC is probably absorbing or depositing on the HAP surface. But we do see a separation of and a rise of the dissipation lines, which is the orange set of lines. And this is very characteristic of a molecule depositing which has a long tail. So we can say that some CPC is depositing, but as soon as we start the wash, we see that CPC is kind of like eluding away with the water wash. But when we are kind of like depositing CPC and Gantrous, we see a sharp change in the frequency, a sharp drop, which means significant amount of material is absorbing onto the surface. And even when we start the water wash, we do not see that material immediately leaving the HAP surface. It eventually leaves, but it takes some time to wash off whatever has deposited. So what is depositing? Is it just CPC? No, it cannot be, because then the dissipation lines would have been separated and we would see a rise. It is the polymer just by itself? No, because the polymer is a long chain polymer that would also raise the dissipation lines. So the only possible conclusion is it's a complex, it's the ionic complex of CPC and Gantrous. And in that complex, neither the polymer nor the CPC now has the extended chain. It's a kind of like a tight coil. So this kind of proves that Gantrous is complexing with CPC and helping it deposit onto the HAP surface, which is our analog for a hard surface, and also retain it much longer than just a CPC solution onto the HAP surface. So in conclusion, Gantrous S polymers interact ionically with CPC or a cationic active and helps deposit them on hard and soft oral tissues. The Gantrous S97 polymer was not seen to inhibit the antimicrobial activity of CPC as determined by minimum inhibitory concentration. And we saw deposition improvement on soft tissues and hard tissues via formation of a reversible ionic complex, which may eventually interpret into maintaining the necessary active level of CPC in mouth over extended time through a sustained release mechanism. That brings me to the end of my slide. Thank you all for your patience. And if you have any questions, I'll be happy to answer it now.
Thank you for your interesting presentation. Do you have any questions? Okay, I have one quick question.
Sure.
I'd like to ask if we can use this polymer to release material other than CPC. For example, can I release growth factor or antibodies?
So this polymer is known to kind of like work with certain oral care actives. For example, triclosan is another active. This polymer works very well. This polymer works with the retention and deposition and retention of certain natural ingredients like natural oils, like ginger oil, or those kind of things, IPMP kind of actives. So there is a range of materials that you can use it for. If you have a certain specific interest, that can definitely be investigated. We were looking into small molecules and small molecule actives, especially cationic actives, because there are two notions that are common in the oral care business. You do not need a deposition aid for cationic actives. They themselves tend to deposit very well, and you may actually hamper the activity of the cationic actives by complexing it with an anionic polymer. So the objective of this work was to show that both of them are probably not true. We can have added advantage if we have a deposition aid like this polymer.
Okay, thanks. Any questions? Okay, thank you, Sako.
Thank you.
And let's move on to the last presentation. The last speaker is Dr. Yamada. Could you share the screen?
Yes. Can you see my screen?
Yeah, good. Go ahead, please.
Okay, thank you. So I'm postdoc at Indian University from last month. Previously, I belonged to Nova South Eastern University for 4 years. All of the results are from NSU. So yeah, let's begin. The title is The Possible Inhibitory Effect of Polypheromonous Gingivalis Derived Phosphoglycerol Dihydroceramide on the Proliferation of Oral Squamous Cell Carcinoma. So, as a background, Polypheromonous gingivalis produce a lot of virulence factors such as LPS and gingivans. It's widely accepted P. gingivalis may have a potential role to promote oral cancer, to support migration and invasion. Recently, it is reported P. gingivalis produce unique family of sphingolipids, and only a limited bacteria can produce this unique sphingolipid called PEDHC and PGDHC. It's unclear about the effect of these sphingolipids on cancer cells. So in this study, we aim to evaluate the potential effect of these sphingolipids on oral squamous cell carcinoma. Next, I mentioned about the sphingolipid metabolism. Among of a lot of sphingolipids, the simplest sphingolipids are called ceramides. Ceramide is the central component of sphingolipid metabolism. Ceramidase is the key enzyme that catalyzes ceramide to sphingosine and fatty acid. There are 3 different ceramidases: acid, neutral, and alkaline. Acid ceramidase is a well-studied enzyme among them, and cancer cells show the upregulated activity of acid ceramidase to protect cell apoptosis and autophagy. It's unclear that if P. gingivalis derived sphingolipids can affect the sphingolipid metabolism. But this paper indicated that human periodontal gingival tissue had less acid ceramidase genes expression to compare with healthy patient. Also, Also here, P. gingivalis can downregulate the expression of acid ceramidase in gingival epithelial cell line OBA9 cells. So our hypothesis is PGDHC and PEDHC can downregulate ceramidases and then accumulate intracellular ceramides, leading to suppress the cancer cell growth. So here I show the material and method. I used OECMR cells and OBA9 cells. I stimulated both cells with PGDHC and PDHC for 24 hours. The methods I used are WST1 assay, scratch assay, transfer migration assay, and real-time PCR, and check the intracellular total ceramide level. So this result shows WST1 assay to check cell proliferation. Regarding to OBA9 cells, both sphingolipids didn't have a significant effect. On the other hand, both PGDHC and PEDHC were suppressed the cell proliferation on OECMR cells. Then we examined the effect of PGDHC and PEDHC on OECMR cells using scratch assay. PGDHC and both different concentration of PEDHC inhibited the cell migration. Furthermore, we performed transfer migration assay. The images below are for higher magnification, and cells are stained with crystal violet. The number of transfer migration cells stimulated with PGDHC and PEDHC were less than control group. So our results indicated both of PGDHC and PDHC inhibited proliferation and migration in OECM1 cells in vitro. So next, to determine if PGDHC and PDHC were involved in the sphingolipid metabolism, we measured total intercellular ceramide and dihydroceramide level in OECMR cells. PGDHC and PEDHC induce the accumulation of dihydroceramides with significant difference, and moreover, PDHC induced the accumulation of ceramide in OECMR cells. Then we evaluated if PGDHC and PDHC affect expression pattern of ceramidases in OECMR cells. Asahi1 expression for acid ceramidase and alkaline ceramidase for ACR2 and 3. ACR2 were significant downregulated by PGDHC and PEDHC. So we showed PGDHC and PEDHC induced accumulation of dihydroceramide and ceramide, and also reduced the expression of ceramidases in OECMR cells. So this is a conclusion for my slides. Interestingly, PGDHC and PDHC have a potential inhibitory effect on oral cancer cell proliferation and migration by regulating sphingolipid metabolism. So PGDC itself has a role to a potential effect to promote oral cancer cells. Thank you so much for the support for this project. Thank you so much for listening. This is all what I'd like to show.
Thank you for informative presentation. Do you have any questions?
Can I just ask a question? Are these two sphingolipids expressed constitutively on Porphyromonas gingivalis, or is the ratio altered in response to things like sensing epithelial cells?
Ratio. Sorry, could you say again about the ratio?
So there are two sphingolipids expressed by Porphyromonas. Are they expressed constitutively, or are they regulated in response to sensing the host?
Actually, I don't know exactly about how they produce those sphingolipids. If it's released to outside or still remain in a bacterial membrane. I'm sorry, but I in periodontal gingival tissue, it seems there are a lot of PGDH are existing, but I'm not sure if it's the samples are biopsy of gingival tissue, so I'm not sure it's PGDH are released to outside or still remain. And also to compare with PDHC, PGDHC are much more in P. gingivalis bacteria existence. Yeah, exists.
Thank you, Dr. Yamada.
Thank you so much.
Thank you all for your presentation. All the presentations scheduled were finished. We have some time left, so we can discuss our research in the rest of the time. I'd like to open up questions and comments from everyone. Are there any questions? Okay. May I ask Dr. Yamada?
Yes.
Okay. As far as I know, I thought PG accelerates onset of cancer. It has been reported that PG was detected more in oral cancer compared with healthy patient. In addition, PG's infection is also known to enhance cancer invasion. And based on these reports, what do you think about the role of PGDHC and PDHC?
So actually, I also think P gingivalis can enhance or support oral cancer, and but it seems it's not so strong to check some review papers, and I also find a few paper PG virus itself doesn't work to oral cancer cells. So to think all of them, probably PGDHC and PDHC has a like a break to other suppression effect, I think. And PGDHC and PDHC are both are ceramide. So in general, ceramide itself are considered to cause apoptosis or photophagy. So I think to think about only ceramide as PGDHC, PGG virus produce, they are just break. But I don't know exactly yet.
Okay,
I think
that's
it's very
interesting.
interesting, yeah.
It's interesting. Thank you.
Thank you.
Do you have any questions or suggestions? Okay, I have one more question. May I ask Dr. Miki Aramiro?
Yes.
Okay. I have one question. According to research, soluble ACE2 decreased in COVID-19 group. This means soluble ACE2 is released under normal condition in healthy people, right?
Correct. Yes. Soluble ACE2 is released in normal healthy patients as well. There is just in normal healthy patients, we still have some gram-negative pathogens, and those are going to activate that ADAM17 to cleave ACE2 into the soluble form, as well as some passive diffusion from the plasma.
Okay. Do you know what function soluble ACE2 has under normal condition?
Under normal conditions, it's still kind of a question mark. With cardiovascular disease, we do see that upregulation of soluble ACE2, but we're still unsure about if it's the cause or the effects related to that cardiovascular disease. So, you know, in normal conditions, it's still kind of unknown about soluble ACE2's function there.
Okay, thanks. It's so interesting.
Thank you so much for your questions.
Thank you. Do you have any questions? Okay, I have last questions. May I ask Dr. Nikojun?
Hi, yeah.
I have one question.
Yeah.
According to your results, the delayed addition of Streptococcus mutans was effective to inhibit the biofilm. And based on this result, in oral cavity, I think probiotics need to exist on the tooth surface before Streptococcus mutans attached in order to inhibit biofilm formation. If so, what is the best way to take probiotics?
So the probiotics with previous research was they don't bind very well to the tooth surface, so it would have to be a repeated use. So like fermented foods sometimes have probiotic organisms in them. Yogurts, just a repeated use. In one of the studies that I did research on with repeated use of, I believe, Lactobacillus paracasei over 2 weeks, it reduced salivary levels of strep mutans. So it's just repeated use.
Okay, thanks. Do you have any questions or suggestions? Okay, it seems I've covered everything. I think it's time to wrap up now. In closing, I'd like to thank the speakers for their contributions and also thank the audience for many suggestions and comments. Yeah. I'd like to conclude this session. Thank you so much. I appreciate that.
Thank you. Thank you, Dr.
Thank you.
Thank you. Thank you all.
Thank you so much.